Hello everybody. This is our plant hormone and carrot tissue culture lab for Plant Cell and Physiology while we're all trying to stay at home. So hopefully this will at least show you what would have been done if we were actually in lab. And this first video is about media preparation. Now I want to welcome Dr. B's students from the University of Delhi's Shahid Rajguru College of Applied Sciences for Women. Dr. B and I have known each other for a long time, and she was my graduate student. I was her PhD mentor. And here's a couple of pictures of her working at the electron microscope while she was collecting data for her PhD dissertation. So for the media prep for this experiment, we're going to use two different hormones. The auxin will be IAA, and the cytokinin will be kinetin. And for each one, we want a final concentration of 1 milligram per milliliter. And the three things we need are the hormones themselves in a powder form, some tubes to put them into, or to prepare the working stocks, and we need some sodium hydroxide and hydrochloric acid. I like to pre-label my tubes, so here they are. They were pre-labeled. And then each one was weighed out, and it just so happened I got 14 milligrams for each one. They were poured into each of the tubes. And then 1 milliliter of either hydrochloric acid for kinetin or sodium hydroxide for the IAA was added to each one. And so it was added. Close the bottle. And I'm just rolling around the hydrochloric acid to make sure all of the powder got to the bottom and shaking it around a bit. These hormones are difficult to get into solution. And so we use the acid or the base, half normal acid or base, to get them into solution. So we add that, and then they were incubated at 60 degrees Celsius, and within about 10 minutes they were dissolved. You'll also notice there's another bottle here. Those are the vitamins, and I had to heat up the vitamins because they have a tendency to precipitate when I store them in the refrigerator. After the hormones were fully solubilized, water was added, distilled water was added, up to 14 milliliters. And with the 14 milligrams of the powdered hormone plus 14 milliliters of water, we end up with our 1 mg per mil working stock. Now, to make the medium, we're going to need MS salts, and this is a mixture of these compounds, major nutrients, which will look familiar to the plant physiology students because these are compounds we tested in our nutrient dropout experiments. We also have micronutrients, and those should look familiar as the different metals that are needed for various reactions to occur. And then we have our vitamins. Now the vitamins, let me back up and just say for the MS salts, I order them from phytotechnology laboratories. You can get them from different sources, but that's where I got mine. And they come pre-mixed. Now for the vitamins, I order those also. I buy those as a 1000x stock. And it has a list of different things in it. It's really has plenty of B vitamins. The medium is called MS, and it stands for Murashige and Skoog. And Murashige was Skoog's student, and the two of them came up with this medium and published it back in 1962, and people have been using it ever since. The other components that we'll need is sucrose and agar. And in both cases, we're going to be using, well, you don't have to use the best quality sucrose, but for doing tissue culture, it's always, you increase your chances of success if you do. So I'll be using ultra pure sucrose rather than sucrose, say, I purchased at a market. And the agar, you'll see it's plant, specifically for plant tissue culture. To make the media, I used a magnetic stirrer, and you can see there's a stir bar right here. You place it into the beaker, and there's just some water in there. And the stir plate has another magnet inside it, which will stir whatever solution that you're trying to make. I think you get the idea. So I'll move on. So the first thing added was the MS salts. So that's 4.33 grams. I'm just trying to get every last bit, and I'll spray it with some distilled water to make sure all of it gets in there. And what I have here on the left is the recipe, and good practice is to write it out in your lab notebook ahead of time, and then every time you add a component, you check it off. Or if something is different and you didn't weigh out the exact amount, you can always change it, make an addendum. Say, like, let's say I added 4.35. I can change that. Next is the sucrose, and you can see it's pelleted, so it's a little easier to get out. And then the vitamins. The vitamins have to be pipetted. And so now we have, with our recipe, of course, we added the MS salts, our sucrose, and our vitamins. Next, the solution needs to be pH'd. This is our pH probe, and we're going to be using a digital pH meter. The probe will be placed into our solution as it stirs. Initially, the pH reading was 4.23. What we want is 5.8. So that means we need to add some sodium hydroxide to raise the pH. When adding any sort of acid or base to your media, you want to do this judiciously. So notice I'm just going to add just like 1 drop. And after that was added, the pH went up to 4.57. So I just continued this. I continued adding drops until we reached 5.82. And that's pretty close to 5.8, and so that's good enough. Next, the agar was added to an Erlenmeyer flask. And then the pH'd solution was poured in. Some people, when they make their media, they prefer to add the agar and then pH it. The agar does change the pH a little bit. In my experience, my tissue cultures have been successful, whether I pH'd it before or after adding the agar. So it's really up to you. It's almost like a style issue of style, whether you choose to pH with or without the agar. Next, the hormones were added, and in this particular case, it should not affect the pH, adding the hormones, because even though kyneton had hydrochloric acid added to it, and the IAA had sodium hydroxide, they were added in equal amounts. So they should titrate one another to neutrality. So with adding those, we've now added everything. So you'll notice all of our components have been added and they're all checked off. And this is the final product. For the second part of our experiment, there were 16 different hormone concentrations that were going to be tested. And that's what you see on the right over here. And so that the process that I just showed you was repeated 16 more times. Now all of it was placed into an autoclave. An autoclave is a gigantic pressure cooker, and it was run on a liquid cycle for 30 minutes. The media, and you'll notice here's some right here. Once it comes out, you'll see it has a yellowish tint to it, but it's relatively clear. The agar, everything has gone into solution during the cooking. And so the media and everything else that was autoclaved, and notice I have water here, H2O. So I have 3 containers of water. Those were also autoclaved, as well as the tubes and such that I would be placing media into. All of these were autoclaved, and then they were put into a laminar flow hood. So that's what we have here, a laminar flow hood. If you were making media for bacteria, or microbiological media, you might pour it on an open bench or put it into tubes on an open bench. But for tissue culture, you need to minimize all sources of contamination. So all other steps will be performed in a sterile environment in a laminar flow hood. So that's the end. The next video will be the preparation of the tissue and placing it on the medium.